Tubex TF Rapid Typhoid Detection. Tubex TF, a user-friendly and reliable rapid in vitro test for detection of typhoid fever infection. Tubex TF is a unique assay format easy to perform. The procedure is completed in a few easy steps and takes less than 10 minutes to get a reliable result for early detection of acute typhoid fever. This instructional film will show you how to perform the Tubex TF assay procedure. For complete information about this product, please read the instructions for use supplied with the product. The Tubex kit contains the following components that will enable you to perform 36 determinations. Tubex reaction well strip, Tubex TF brown reagent, Tubex TF blue reagent, Tubex TF negative control, Tubex TF positive control, Tubex colored sticker, Tubex sealing tape, Tubex TF certificate. Instructions for use. To perform the Tubex TF assay properly, you'll need the Tubex color scale, a magnetic stand for separation and scoring of results. You'll also need common lab equipment such as a precision pipette and a timer. A vortex for mixing the reagents is recommended. Labeling the reagent bottles with the Tubex colored sticker is also recommended. Place stickers both on the cap and on the bottle. Vortex or shake thoroughly all the Tubex TF reagents. Check visually that all sediments have dispersed into solution. Insufficient mixing may lead to false results due to wrong particle concentrations. Before you start, bring all the reagents to room temperature. In our example, we'll be using the well number 1 for the negative control and the well number 2 for the positive control. We recommend that positive and negative controls are made at every test occasion. Wells 3 to 6 will be used with different samples. Place the reaction well strip upright on the bench. Add 45 microliters Tubex TF brown reagent to each well to be used. Add 45 microliters Tubex TF negative control to the appropriate well. Mix 10 times. Avoid foaming. Add 45 microliters Tubex TF positive control to the appropriate well. Mix 10 times. Avoid foaming. Vortex each sample prior to use. Add 45 microliter samples to the appropriate wells. Mix 10 times. Avoid foaming. Repeat the procedure using a new pipette tip for each new patient sample. Incubate the reaction well strip on the bench for 2 minutes. Add 90 microliters of Tubex TF blue reagent to each well. Cover the reaction well strip using the Tubex sealing tape. To prevent any leakage, press the tape firmly, making sure that it adheres to the surface. Leakage may lead to false results. Hold the reaction well strip at one end using your thumb and forefinger. Tilt it 90 degrees in order to expose a maximum well surface for the mixture. Move it fairly rapidly backwards and forwards for 2 minutes. Make sure that the contents flow across the entire exposed surface of the well. Shaking them incorrectly may produce false results. Place the reaction well strip on the color scale as far to the left as possible. Allow 5 minutes for separation to obtain a clear supernatant. Now you're ready to read and score the results. The reading should be performed within 30 minutes after separation. Move the reaction well strip across the color scale. Read and score the results by comparing the color of each supernatant with the Tubex color scale. The interpretation should focus on the detection of any blue tone in the supernatant, especially in the lower scores. The first well, which contains the negative control, scores 0. The second well, which contains the positive control, scores 10. The third well scores 4. The fourth well returns a score of 6. The fifth well scores 8. And the sixth and final well of patient sample scores 10. A score of 2 or less than 2 does not indicate current typhoid fever infection. A score of 3 is an inconclusive score. Please repeat the analysis. A score of 4 is a weak positive. It's an indication of current typhoid fever infection. A score of 6 to 10 is positive. It's a strong indication of current typhoid fever infection. Tubex TF results should be interpreted in conjunction with all available clinical information. If you follow the procedure using Tubex TF as described in this instructional film, you'll get reliable results in the detection of acute typhoid fever.